students, I'm Dr. Nidhi Sharma, Associate Professor, Zoology, p.g.govt.college, Jhalawar. Dear student, I shall talk to you in this video about the skeletal muscle arrangement in continuation of my earlier part 1 video of muscular system. A single muscle cell is a muscle fiber. Fibers are made up of myofibrils. Myofibrils are made up of thick and thin filaments. The sarcolemma is the muscle cell membrane. The sarcoplasm is the muscle cell cytoplasm. As shown in this figure, you can see the sarcolemma, which is depicted by the blue cover, and in the myofibril, the muscle fiber, you can see the sarcoplasm. You can also see the nucleus in the sarcoplasm. Orange colored big mitochondria are also seen. Here in this diagram, one myofibril has been cut open, and wherein you can see one sarcomere. Sarcomere is from one Z disc to the next Z disc. The cut open sarcomere shows the arrangement of thin and thick filaments. The skeletal muscle cells. Myofibrils are striated. The striations are due to arrangement of thick and thin filaments, seen as alternating areas of light and dark bands. The length of each myofibril is divided into repeating units called sarcomeres. A sarcomere is the functional unit of skeletal muscle. Hello students, in this slide you can see the arrangement in a sarcomere. A sarcomere has different sections. The first section, we can say the sarcomere starts from one Z disc to the next Z disc as shown in the figure. Then you can see the H zone. The H zone contains an A band. The arrangement of the thick and the thin filaments are shown in this figure. The I bands are also shown in this figure. This is in this figure you can see an elaborated sarcomere wherein you can see a myofibril, and in the second figure you can see details of filaments and discs and Z discs. The myofibril, as shown in this figure, starts with a zigzag Z disc from one end to the zigzag disc, a Z disc in the other end. The thin filaments are depicted as grey colored, and then The blue line shows the M line. The thick filaments and the thin filaments alternate each other. Here in this, the I band, after the I band, you can see the zone of overlap wherein the H zone is present. Again, it is followed by another zone of overlap, and this depicts the A band. The sarcomere structure. Sarcomere exists from Z line to Z line. A band is dark middle band. It is overlapping thick and thin filaments. I band. Ends of A band, thin filaments only, make up the I band. Z line is in the middle if of the I band. Myosin filaments are held to the Z line by titin proteins. Here you can see the microscopic anatomy of a skeleton muscle fiber, wherein you can very clearly see the sarcomere from one Z disc to another Z disc. In the middle, you can see the M line. The arrows point to the I band, which are comprised of thin filaments in the first circle. The second circle shows the edge zone with thick filaments only. The third circle shows the M line, which has thick filaments linked by accessory proteins. And the fourth circle depicts the outer edge of A band, which has thick and thin filaments overlapping. The thick filament structure. Thick filaments are composed of many myosin molecules. Each myosin molecule has a tail region and two globular heads, which are known as cross bridges. The thin filament structure. The thin filament is composed of actin protein. Two strands of globular actin molecules are twisted into a helix. Here you can see troponin complex depicted by yellow color and tropomyosin, which is the twisted brown color, and then the spherical rounded actin in blue color. This structure shows the sarcomere, and again there's an elaborate structure depicting the thin and the thick filament showing the globules, the Z disc, the titin filaments also. Now we will study some specialized organelles of skeleton muscle in the next part of the muscular of the skeletal muscle.